Over 90% of new chemical entities fail clinical trials. This is not because they're ineffective, this is because they can't dissolve. Poor aqueous solubility. And we have been predicting this all wrong.
My Aqueous model gets an R-squared of 0.74 on aqueous solubility prediction, a 13% improvement over the original ESOL. And using only 12 descriptors, this is much better than what we see in literature. However, when you ask the model why it's performing that way, you get the same rank list every single time, LogP, TPSA, with no chemical context.
So I asked, what if these rankings aren't universal? What if it changes across chemical contexts? I stratified 9,982 compounds in the Aqueous dataset, using Lipinski's rule of 5 into drug-like and non-drug-like compounds. And then I calculated trap values for each of them, taking the ratios, to finally arrive at the CPTS metric, which was the bin-specific importance divided by the global importance. And the pattern was undeniable. In drug-like chemical space, LogP showed an importance of 40%, but in non-drug-like space, 75%. And this is with a p-value of 10 to the power negative 101. So this isn't just a random artifact, this is an important distinction in the way that solubility works. And the great thing is, this isn't limited to LogP. 9 descriptors showed statistically significant differences across chemical contexts. Number of rotatable bonds, aromatic rings, and fraction sp3 all showed chemically dependent insights. And these aren't just random artifacts. All statistically validated and independently tested on ESOL, these are actual real chemical rules governing solubility. From this, we derived 5 actionable rules for drug design. It talks about when to prioritize lipophilicity, when to constrain molecular weight, and when hydrogen bonding matters the most. This is important because it gives a clear roadmap for Indian pharmaceuticals and biotech startups from day one, giving them time to save money, energy, resources, and also increase success. The amazing thing is, this isn't limited to just solubility. Permeability, toxicity, binding affinity, all of these are context dependent. So now, we not only know the what, but we also know the why. And this lets us design better drugs.